Well, good evening, everyone, and welcome to the Stockmen's Association Seedstock Council virtual webinar. I think we'll have a few people still popping on, but we sure appreciate people taking time out of their evening to join us for this special event. But I think we'll go ahead and start some introductions and get on our way with the evening's program. For those of you who don't know me, my name is Julie Ellingson. I work for the North Dakota Stockmen's Association here on staff. And I, like you, am an Angus seed stock producer. And so looking forward to hearing about the presentations tonight. The Stockmen's Association, as many of you know, is a 96-year-old beef cattle trade organization. We prided ourselves on uniting, protecting, promoting, educating, which we're here about tonight, and serving the beef cattle industry. And so I think tonight will be a wonderful example of that educational component. Looking forward to, as I mentioned, a great webinar. I'd be remiss if I didn't recognize our sponsor, the North Dakota Stockmen's Foundation, a 501c3 charitable group with a connection to the association, but a separate charitable entity, is a sponsor of tonight's program. Again, works in areas, as I'm sure you're well aware, things like scholarships, disaster relief, research, as well as a strong supporter of professional development and educational programming. And so thanks to the foundation for its continued support. Before we get started, and it looks like most all have already muted their microphones, just so we don't have any background noise or such, just as a reminder to do that, or we might mute you if there is some background noise when it's time to talk. And if you'd like to, just be sure to click your button off so you can be heard and can ask questions or offer some input. We'll have the opportunity to ask some questions at the conclusion of each of the speakers. And so again, you can do that either by unmuting your button and then posing that question directly, or if you feel more comfortable, you can type that question in the chat and we'll help make sure that our speakers have access to that and know what your question is. Or if you prefer, you also can email Elizabeth here at ehassenwinkel@ndstockmen.org. And so we will watch for questions in all three of those places. And so with no further ado, I would turn, I guess, the floor over to my friend and colleague, Elizabeth Hassenwinkel. Elizabeth, as many of you know, I probably had a chance to work with her real closely as a longtime member of the Stockmen's Association team. Elizabeth has been gone from us for a little while, exploring another opportunity, and I'm very excited to report that she'll be coming back home to the Stockmen's here starting next month. And so grateful to have her back as our communications director here shortly, and very grateful for her support with some projects even in the interim. And I know that she has a strong commitment to the association and beef cattle production in North Dakota. So to talk a little bit more about the Seedstock Council, which is one of her responsibilities as a staff liaison when she's back on staff, we'll let you hear from Elizabeth.
Well, good evening, everyone. As Julie said, my name is Elizabeth Hassenwinkel. I was the communications director for a very long time, took a little leave, and I'm now going to come back here next month. And so I'm excited to be back and working for the beef producers of North Dakota. The Seedstock Council, as she also said, is one of my responsibilities as the communications director. And so we are going to promote the seedstock industry. That's what the mission of the Seedstock Council is. And do education for both producers and their customers through planned programs, printed information, legislation, and other feasible means. Tonight, we're going to go through what the Seedstock Association is, and then we're going to talk about everybody that's kind of involved with the Seedstock Association. So I just flashed across your screen here. You should all see it as long as it's working correctly. The current members of the Seedstock Council. There are several members whose terms will be coming up soon. And so if you have any interest in serving, please reach out to our office and we'll get you in touch or reach out to your respective breed association. I'm going to turn over the floor to our current chairman, and he's been our longtime chairman, Travis Bruner, and he's going to introduce our first speaker.
Yes. Thanks, Elizabeth. As they all said, one of the objectives of the Seedstock Council has been to bring continuing education programs to the producers. This webinar has become an annual event and was actually born during the pandemic and was a way to bring industry together without having the long drives and late nights of travel to do it. One of the topics of the Seedstock Council pegged a priority last year was bull fertility. And it started with conversation with Dr. Bob Sager of Medicine Creek Bovine Health Clinic at last year's webinar. We decided to continue to take a deeper dive into the topic and help answer the question of why do some bulls get cows pregnant more efficiently? And so tonight we're pleased to welcome Dr. Lacey Quayle and Dr. Solo Zoka to explore the key factors driving bull fertility, the role of semen quality, and new tools helping us develop fertility markers. Dr. Quayle is the NDSU Livestock Management Extension Specialist. As a graduate student, her research focused on the role of estradiol in pregnancy maintenance in beef cattle, as well as improving reproductive efficiency and profitability of beef operations through the use of reproductive technologies. In her current role at the North Central Research Extension Center, she develops and delivers livestock management programs across North Dakota while collaborating with extension specialists and agents, research faculty, livestock organizations, and producers. Dr. Zoka comes to us from the University of Tennessee, where he is the assistant professor and extension beef cattle specialist. His work focuses on advancing reproductive efficiency in beef cattle by integrating applied and translational research with producer-driven extension. His work has examined bull fertility from multiple angles, including the identification of sperm proteins and metabolites associated with fertility and the impacts of nutrition management and herd health on pregnancy establishment and loss. So if everybody could welcome Dr. Quayle and Dr. Zoka, and we sure look forward to the information that they're going to bring us tonight.
All right, I'm going to go ahead and share my screen here. All right, just want to double check that we're all seeing the same thing. We're good?
Yes, very good, Lacey.
Okay, perfect. All right. Well, thank you, Mr. Bruno, for that introduction. And I think I can speak for both Salo and myself that we're excited about the invitation to be here tonight and just the discussion about male fertility in our beef herds. And I think this is a great opportunity to spark some of that. So just to go ahead and get us started tonight. I thought I'd start us off easy with the question of what is a bull's purpose in our beef herds, right? And I think we'd all agree that the bull's purpose is to produce pregnancies, right? Not necessarily produce sperm, but that's a necessary part of the responsibility to produce pregnancies. And I also think we could probably agree that a single bull in the traditional herd, in the traditional sense, a single bull has a much greater genetic impact than, say, a single cow, right? And that we have natural service sires that are being mated to several females during a season, whereas a single female, ignoring operations and flushing and things like that, in a traditional herd, a single female is just going to pass on genetics to one calf, right? This gets amplified when we think about artificial insemination, particularly. On top of that, we know from some national data that over 90% of mature cows and 76% of heifers are exposed to natural service sires each year. And so this really puts the focus on male fertility, right? And how we need to make that a priority when it comes to productivity and profitability in our beef herds. So I wanted to start tonight just by going through kind of a simple timeline of what we consider to be bull development, right? Let's say a bull calf is born mid-March, maybe that calf is weaned mid-October, where they go through a development period after that before reaching their first breeding season that next spring. And so that development period is something that we focus on a lot, right? Because we want them to be successful in our beef herds for long periods of time. And it's during that development period where they reach puberty. And we work to manage them in a way that they reach puberty at an appropriate age, right? And so the standards for puberty might look a little different than maybe what you're used to as far as the requirements of a breeding soundness exam. But just to kind of introduce those here quickly, as a bull attains puberty, we see a scrotal circumference of 28 centimeters. They need to produce at least 50 million sperm cells and have at least 10% progressive motility. As they transition then to being a mature sire, this might look a little bit more familiar as far as the requirements for a breeding soundness exam. Depending on the age of that sire, they'll need at least a 30 centimeter scrotal circumference with at least 70% normal cells that have at least 30% progressive motility. And so I want to highlight here that the transition from puberty to being a mature sire is not it's not always black and white, right? It's a gradual process that occurs over time, and that's something that we need to focus on in this fertility discussion. So I think if we want to know how to improve or manage male fertility, we should know the ins and outs of sperm production in general. And so I want to go through that here tonight, just briefly. But in the test is where sperm are produced, but more specifically, we have these lobules, right? And there's these squiggly lines in there that are more specifically termed seminiferous tubules, and that's specifically where sperm cells are produced. And I like to think of this more like a production warehouse, right? Where you have the production floor, you have the packaging floor, and you have the delivery floor. And very similarly, we have sperm cells that are produced, and then they are kind of shuttled into this middle area before they enter the epididymis, where they go through their final maturation. And so in that way, I kind of like to think about it like a warehouse a little bit, and I'll probably use that analogy as we move forward. So I don't want to belabor this by any means, but I put this here to show that sperm production is a very complicated process, and part of the process is devoted to multiplying the number of cells. So the top part of kind of this flow chart here is showing how we have spermatogenic stem cells that are used to produce and multiply the number of sperm cells produced. The second part of this process, down below the dotted line, is responsible for increasing the genetic diversity of sperm cells, so that the billions of sperm cells that are produced each day, not two of them are the same. So I really like this figure because it shows everything that's happening in this little lobule, in this little squiggly line. This is exactly what's happening. And to make maybe this even harder to understand or even harder to control from a management perspective is that this complicated process is also continuous, meaning that every segment of this tubule contains every part of this production process. And simultaneously, all these parts of the production process are occurring at any given part of that part of the testes. So you'll notice that there's several different types of cells that are involved, and I've matched them to where they're at in the part of the process. And so some of these look like sperm cells, right? That you might recognize as having kind of that elongated head and a tail. So that might be familiar and look like a sperm cell, but there's several others that don't, right? We have ones that are just circular and don't really look like much anything different than a regular cell. And so in this way, we think of this like a freshman matures to a sophomore, matures to a junior, and finally graduates as a senior. There's some similarity here in that these cells down here eventually mature to become these type of cells, which eventually mature to become this cell, and then finally they get to look like a sperm cell and mature, and those are what are actually released for the packaging and the delivery parts of the warehouse. So this whole production process takes approximately 61 days from start to finish before these sperm cells are released for their final maturation. So kind of the take home from those few pictures and that process is just that puberty and sperm production in itself is a gradual process, and it relies on several different types of cells and several hormones that are required to make that all happen and happen appropriately. The second thing is that sperm cells need to mature before they are capable of fertilizing an egg cell and producing a viable pregnancy. So these 2 points kind of combined mean that an insult to any point of this production process can impact future fertility. So not only does an insult to, let's say, heat stress or nutritionally, a nutritional stress, not only does that impact the phase in production that impacts cells that actually look like sperm, but that same insult impacts the cells that are at a much immature stage of this production cycle. And so that's why that one incident can cause fertility issues for weeks or even months at a time. And because sperm production is continuous, a sire can go back and forth in fertility status throughout his life. So from fertile to subfertile and back to fertile again. And so when we think about improving spermatogenesis, right, that was one of the thoughts of this topic going forward for this webinar, that really means managing our sires to avoid what we already know will decrease fertility, which I hope to talk a little bit about tonight. And then also in the future, looking for ways that we can identify sires that have a greater fertility potential, which Dr. Zoka will focus on. So if we know that a sire can go from fertile to subfertile back to fertile throughout their lifetime, it's hard to know exactly where we're at in that process, right? And one of the best ways that we have to do that is breeding soundness exams. And I don't want to belabor this either, because I think it's something that we're all pretty familiar with, but it is the best snapshot of fertility that we have across the industry. And it contains 3 unique parts, right? It's not only just a semen analysis, but it's also a physical exam and measuring scrotal circumference. And a bull has minimum requirements of all 3 of those that they need to meet before being considered a satisfactory potential breeder. It's inevitable that some sires will not pass their BSE and they'll be classified as unsatisfactory. So in fact, out of 6 studies that pulled together, they collected over 8,000 bulls. 24% of them were classified as unsatisfactory. And so that doesn't mean that they're sterile or infertile, but they did fail to meet the requirement in at least one of those 3 categories. And so in some regard, we can consider them subfertile. So if we weren't to have done a BSE, or if we, chose not to do a BSE on those on those sires, and those subfertile males were turned out with a group of females, what impact might that have? And Sal and I both like to say that a subfertile sire will get females pregnant, but they'll just likely take longer to do that. And so I want to go through an example just really quickly of a few scenarios that show kind of the changes in productivity and profitability that subfertility can have in our sires. So this is an example of at what point, if we say, for an example, in a 100-cow herd, at what point of the breeding season do those females get pregnant? And so you'll see at the end of the breeding season, we have 95% of our females getting pregnant, but it varies on when they got pregnant. So whether they got pregnant in the first, second, or third cycles. And the variation isn't large here. We have 60% in the first cycle, down to 50, and then down to 45. And the third cycle pregnancy rates vary anywhere from 10 to 20%. So just kind of keeping that in mind, and we'll go through how that changes productivity. Let's say at weaning, our steer calves are weaning a little heavier than our heifers. And of course, based off of when they were born in the calving season, that's going to influence their weaning weight. I used an average of 2.2 pounds per day that they'd be gaining on the cow here prior to weaning, and just kind of accounted for that difference in age going forward and what their weaning weight might be. So some pretty easy multiplication, we can use those numbers to kind of determine the number of pounds weaned in each cycle, right? So in each time point of the calving season, the amount of productivity that we have on a pounds weaned basis. And if we go through and we add those all up, By the time we get to the bottom, we're at about an 1,100 pound difference in weaning weight by taking our first cycle pregnancy rates down to 45% and upping our third cycle pregnancy rates to 20%. When we start to put a dollar amount to that, looking back on some sale reports from last week, it's pretty easy to find a price point for those weights and do the same type of math again. And by the time we're done with it, we're looking at a difference of over $3,500 from our very last scenario, with 20% of cows getting pregnant in the very last cycle, and 45% of cows getting pregnant in the first heat cycle. So $3,500 in difference in revenue then, just based off of when females are getting pregnant throughout the breeding season, as kind of a reflection of male subfertility. And it's true that BSEs definitely have a cost, right? Let's say that in this 100 cow herd, we use 5 bulls, 1 bull to every 20 females, and it costs $50 to have a breeding soundness exam done. That $250 is paid for pretty quickly, just in scenario 2, right? And if you remember, that still had 60% of cows calving in the very first bit of the calving season, and we only shifted things about 5% in these last two cycles. So definitely have a cost, but subfertility also has a cost. And while we can work to manage reproductive potential in our sires the best to our ability, a BSE is our absolute best snapshot of that sire's fertility at any given point in time. Okay, so back to those management factors that we talked about to hopefully manage fertility to the best of our ability, right? And in that 61-day time frame, what can impact a sire's reproductive ability? Well, there's a whole host of factors that can do that and can impact a sire's ability to produce quality semen. From the production part of the factory, improper nutrition or poor feed quality, changes to scrotal temperature, whether that be too hot or too cold, scrotal frostbite, and maybe some kind of more long-term injury from that as far as scar tissue goes, or even getting stepped on, and disease. All of those can greatly decrease a sire's ability to produce quality sperm cells. A little bit further down the line, when we think about something maybe more applicable to an AI situation, how that semen is collected, processed, and packaged, and then handled by us and thawed, can all impact the sperm cell's ability to fertilize an egg cell. So there's a few of those factors that, especially on the sperm production kind of side of things, that also influence a sire's marketability, and body condition is one of them. And the University of Florida has a bull test every year, and they actually tried to put a number to this, right? And so they looked at the correlation between a sire's final body weight and what they brought at the sale. And this number, 0.46, that's considered a moderate correlation between those two things. So as final body weight increased, sale price also increased. But we also know that body weight typically comes with body condition, right? Those two things tend to increase together. And some work out of Canada, some research there has actually done a great job at pinpointing how body condition score influences a sire's ability to pass a BSE. So in this study, they collected over 1,600 bulls, and what was unique about this study is that every single one of those bulls passed the physical requirements of a BSE. So that means the scrotal circumference requirements and the physical exam. So the only part left was the semen analysis. And of the bulls, they ranged from a body condition score 4 to a body condition score 8. And you'll notice here the percentages of the percent of those bulls that passed a BSE. And so while the thinner bulls weren't necessarily quite as likely to pass a BSE compared to those in maybe more moderate condition or above, slightly above moderate condition here, the real detriment was in bulls that were heavily conditioned. And only 48% of those bulls passed a BSE. And this probably isn't new information, right, to you guys. And what this information did do, even though it might not be new to us tonight, was that it opened the door for further research that wanted to look at how exactly and at what point and to what degree changes in nutrition and nutritional status impacts sperm production. So to kind of lead into some of that research, heavier bulls or heavier conditioned bulls also tend to have greater amounts of subcutaneous fat, right? Back fat thickness tends to be greater in those sires. So a study out of Georgia had over 700 bulls on test, and they were all fed the same diet. So like other species, there are some animals and individuals that it's just easier to, or harder to keep condition on than others, right? They just inherently do that easier or harder than maybe another sire. And so this study was a great example of how the accumulation of back fat without nutritionally pushing for it affected sperm production. And so they took these sires and they measured the back fat thickness, and then they sorted them into whether they were either in the bottom 10%, they averaged 0.2 of an inch of back fat, or the top 10%. Those sires that were in that category averaged 0.5 inch of back fat, and then the middle 80%, which averaged 0.3 of an inch of back fat. And so they had those three groups of sires that they then were able to compare to each other when it comes to the percent of normal sperm cells that they were able to produce, and then also the percent of sperm cells with abnormalities, both primary and secondary. So primary abnormalities being those that aren't thought to improve with time necessarily, whereas secondary abnormalities might, the thought is that they might improve with time and may pass later. So when they looked at the percentage of normal cells, there wasn't any difference, and both well exceeded 80% here between the bulls in the bottom 10% of back fat thickness and then that middle 80%. So they had equal percent of normal sperm cells in those two groups. The bulls with slightly more back fat, they still had 80% normal cells, but that was slightly lower than the other two groups. When it comes to the percent of abnormal cells, those bulls that had greater back fat thickness did also have fewer normal cells and also had greater percentage of abnormalities, both primary and secondary. But these bulls, as you think about it, being that they had 80% of normal cells, they very well may be classified as satisfactory breeders, barring any issues with the other two aspects of the BSE, right? Also in this study, they kind of looked at the probability then based off of if you measured back fat thickness, the probability of failure. And so while those bulls still had 80% normal cells at 0.5 inch of back fat, as back fat increases, and this is in centimeters, but this would be about 0.6 of an inch of back fat, somewhere up in here would be closer to 0.8. The probability just kind of increases in a pretty continuous manner as far as the closer we're getting to failure, or the probability of failure increases. Okay, so I mentioned that even though those bulls had a few more abnormalities, they also had 80% of normal cells, right? And as we know, it takes both a viable sperm cell and a viable egg cell to produce a developing embryo. So what we're learning from the research that's been done on male fertility is that a sperm cell contributes a lot more than what we thought just a few decades ago. And so even though those heavier conditioned bulls with more back fat still produce normal sperm, are there any other consequences maybe a little bit further down the road after fertilization occurs? And so this study kind of sought out and was designed to answer some of those questions when it comes to the embryo production side of things. And so they took 8 bulls that they passed a BSE, and then they were put in either one of 2 diets, and they were fed individually for 67 days. And so half the bulls went into a high gain diet, and they were fed to gain 4 pounds a day, and the other half were fed a maintenance diet, so just to maintain their weight. After that 67 days, they were collected again, and then those semen samples were analyzed both fresh and they were frozen to be analyzed again after they were thawed. And then they were all used to produce embryos in vitro. So when they started looking at percent of viable sperm from the high gain bulls and the bulls that were fed that maintenance diet for 67 days, and I should mention too that 67 days is it's long enough to go through an entire spermatogenic cycle, right? I mentioned how that takes 61 days. So they were fed long enough in this diet to go through an entire spermatogenic cycle. And so this figure just shows that there wasn't any difference in the percent of normal cells from bulls in either group when the sample was analyzed fresh. When those semen samples were analyzed after they had been frozen and thawed, there was a trend to start seeing that those high gain bulls had fewer normal cells after freezing and then thawing. But that was only one part of this project. Again, they wanted to look at embryo development and what that might, how that might change. And when it comes to embryo development, we also know that that's another complicated process, right? We have fertilization occurring, and then just a couple days later is our first cell division. And then by day 7, we have the embryos that are typically transferred, right? If they're collected from donors or being transferred into recipients, typically those are day 7 embryos. And then there's a few more changes that happen in the next week to 10 days that are pretty crucial. And then 265 days later, you have a live calf born, right? So they focused on these first 7 days, knowing that there's a pretty big drop in kind of viability in that first week of pregnancy. And so they looked at how many made it to this stage and made it to that very first cell division. But then of those, they wanted to know how many made it to that kind of transferable stage. And so what they found was the high gain bulls actually produced fewer embryos from their semen samples compared to bulls that were fed that maintenance diet. And so this study just in general shows that there are impacts to more than just general sperm production when it comes to nutritional management. So yes, they looked at percent normal cells, but also looking at how those cells create embryos was a pretty key finding. And so with a bull's sole responsibility to produce viable pregnancies in the herd, it's not enough that he's producing just normal sperm cells, right? That's another layer to the fertility question is, are they able to produce viable pregnancies? And that's something that Dr. Zoka will talk about more about.
Thanks, Lacey. And I mean, if you allow me, I just want to make one more point on this last section you're talking about, Lacey. So very, very intriguing about that study is like you show very minimal changes in semen quality, right? But we will start seeing detrimental impact on the embryo, which bring us to that, yeah, we have normal sperm, but there is something intrinsic with even within those normal sperm that is impacting. So if we go back all the way to that bull development young sire study that you showed, yeah, they're passing BSC, but there is a good chance that they're still producing sperm that will not generate good pregnancies, right? Even though they're passing. So with that, I think it leads really nicely into this portion of the presentation that I'm going to be talking about related to compensable and uncompensable traits. Can you go back one slide, Lacey? Sorry. Okay. That leads really nicely into here when we talk about and I'm going to explain a little bit what are these compensable and uncompensable, right? Because if you think about, there is differences in the quantity and quality of that sperm, and both are actually important for fertility, especially when we're thinking about artificial insemination, right? If we think about natural surrogacy, quantity of sperm, so the amount of sperm being put in the female is far more than what we think would be the minimum. But in an insemination standpoint, we can go very, very low, right? For example, normal doses for sex semen ranges from 2 to 4 million sperm inseminated, and for conventional semen, somewhere between 10 and 40 million sperm inseminated, right? So there is, if you look here, there's several studies that look into that relationship. Okay, if as we increase the insemination dose, what happened with the fertility here, and then we have the dashed line here on this figure showing the optimum female fertility there, right? So as we move to the right of the graph, we have more sperm being inseminated. As we move up in the graph, there's more females getting pregnant until we reach that optimal female fertility. So first, I want to talk about what is the compensable traits, right? So the compensable traits basically are, as we increase the insemination dose, we keep improving fertility until we reach the maximum fertility or the optimal fertility for the female population. And I have two examples here, right? Both A and B, and both of them reach that optimal female fertility. However, if we look at here, they reach that at different insemination doses, and that's critical here, right? So hopefully, and this is a hypothetical scenario, right? So this is what we consider they have compensable traits, but we overcome these compensable traits by increasing insemination doses, right? So, but we also have what we call uncompensable traits, and then we have here on this figure two bulls examples too that we're going to be talking about the uncompensable. So bull C and bull D. So even though we continue to increase that insemination dose, we never reach the optimal female fertility. And a good way to explain, or another way to put this, thinking about a competition, right? So when we're thinking about the compensable ones, the bad sperm does not compete with the good sperm. So as you increase the total number of sperm, this, because we are not competing between the good and the good can keep getting better and better and getting more cows pregnant. However, on the uncompensable side, the issue is it's competitive, right? So both of those, the good and the bad sperm, can actually reach the site of fertilization and fertilize the oocyte, but the bad sperm will not develop into a viable embryo, right? So that's the biggest difference there. So, or have other issues that don't let that embryo start developing. So as we move on, and here's a study I did during my master's, we actually look at a commercially available doses here from 10 to 40 million insemination sperm per insemination in this study. And we did that actually in Brazil. We bred about 6,000 cows, 5,000 to 6,000 cows in this study here, and we saw no difference. And the reason we had 20 million sperm per dose, and the only difference between those two doses was the color of the straw. And whoever was inseminated was blind for that the treatment we had. And if we look at the difference we had between the 220 insemination doses, is actually the same as we had between the 10 and 40. What this tells us is the commercially available doses that our AI industry puts out and sell to us actually is sufficient to overcome what we were talking about, the compensable traits. Okay, so we have worked through it to make sure we can overcome it. And this is all with conventional semen. However, digging further into this study, next slide, Lacey. We actually had 5 bulls within that study, and we start diving more into the results. We actually had 2 bulls out of those 5 bulls that had below average fertility or considered low fertility, especially compared to that bull A and B, right? So the C and D, the green bar there are the bulls that were low fertility, and the A and B were the high fertility bulls, and the D bull is actually the average fertility. And this is not new for us that we have variation in fertility among bulls, but we select those bulls that we were expecting not to find it, but we still found differences. And we look at semen quality for those bulls in that study, and we were not really able to, like traditional semen quality, identify why those bulls were different. And this other table here is some of the data I've been collecting here since I started in Tennessee. How about 1,600 or almost 1,700 inseminations with average pregnancy rate of 58.1. All those bulls are Angus bulls. They are bred either to purebred Angus cows or crossbred cows. And we can see the range we're seeing in pregnancy rates. Of course, some bulls we have limited observations, but if we look at the bottom 5 or 6 bulls, that 5 bulls, we have about 53.1% pregnancy rates with 475 inseminations. But if we look at the top 4 bulls, we have similar numbers of inseminations. We have almost 64%. So almost a 10% difference there, approximately a 10% difference. So we are in my lab and in some of the studies that I've been doing, we're trying to understand why we have that and how can we work behind the scenes so we have less of these bulls in the bottom end getting out commercial, right? And even going further into, okay, is there something we can do on a breeding sounds exam that we can help us detect these bulls that are potentially to be low fertility? And this level of pregnancy, it will be associated to that uncompensable trait, right? Because all these bulls pass that minimum criteria that we needed for them to be out there, right? So all of them had at least a minimum level of fertility. And then we still see differences in the field fertility. So when we talk about developing these new fertility markers, there's several things we need to start thinking, or there's several ways we can do this. But first here, really, if we start thinking about this, when we try to develop these new fertility markers, we're really trying to differentiate those bulls that have high pregnancy rates versus low pregnancy rates, or embryo producing, like we saw on SIG4 data that Lacey showed us there. How can we actually separate those before we actually use those animals, right? So the uncompensable traits, that's actually what we're trying to trigger, right? Because I showed you, really, we got a handle on that compensable trait. So at first we thought, okay, if that sperm is dead, it's bad. If alive, is good, right? But we start keeping digging into this, and then we notice that, well, not necessarily all live sperm are good. There's actually some that are good and some that are bad. So when we put this together, what we come up with is that, okay, what do we currently do? So above this yellow line there, which is our diagnostic adoption, is what we do today to separate this fertilities, right? So we use motility, which is a fairly low bar that they need to cross, right? That's 30%. Even when thinking about artificial insemination, right? They have to be 60% to be frozen, but still we're talking about 30% the post-op. Then we look into morphology. It's a little higher bar, right? It's 70%. But we know even within that 70%, we still have bad sperm in there. And we saw it with the fat bulls, right? So that's a really good model for us to understand, okay, there's more things. So what really we're trying to do now is create new biomarkers that we can separate this potentially fertile into the most fertile and the infertile population within that inseminate, or that ejaculate. So when you're thinking about actually fertility, it's actually multifactorial, right? And it really depends on several events, or a multitude of events that must occur in an orderly fashion for a viable pregnancy to establish it. Very similar to what Lacey was showing us, right? It needs to fertilize, and there's all those stages of cell divisions and go on, and it's especially early on, before that day 28 approximately, is where most of that, actually we know even before that, probably earlier than day 21, 22 is where most of those pregnancies will be lost. So identifying things where it's hard for us to actually go there and measure pregnancy. So identifying things that we can help us improve something that we can really measure that early. So when you start thinking multifactorial, right? What do I mean about fertility is multifactorial? So let's think about this figure here, like this square, this cube here. Let's think about right in the center of this cube is what we would consider the optimal fertility. And we have all these little arms pulling it and trying to get it away from the middle there, which would cause that animal to change from optimal fertility, right? So what do we know? And we just talked about this, right? Progressive motility and normal morphology, right? We have a certain level that we know if you're below that, that arm start pulling away from optimal fertility towards subfertility. But in reality, I have here what 3, 6, 8 little arms there, but we don't fully understand. Is that actually 4? Is that 10 or is that 200 that actually are important markers there for that fertility? That's where the multifactorial point come from. We also understand DNA fragmentation. So the DNA within that sperm that is being delivered to the oocyte needs to be intact, right? So as a one string. As we start having more percentage of more sperm with defective DNA, we know our chances of pregnancy start getting low. It is not a standard measurement within our AI industry, and the reason for that, and I have talked with some of the research folks within our AI industry, and they show me the data that most of the time, all their bulls is already above the minimum. Okay, so it's not really an issue with the bulls that they currently have. Every now and then, if they find something that is bad, they may go back and try to check it and start doing some more diagnostic. But in reality, we don't see a whole lot of issues in our bull. And in part of that is because we select animals to be more fertile. We've been selecting for it. A good example of where this is relevant and it's important is actually in humans. In humans, we know that DNA fragmentation is a big component of subfertility and infertility. Most actually in humans, we don't talk about subfertility, talk about infertility. And then when we get high DNA fragmentation, we start having infertility in males. But there's other pieces to this puzzle that we know that they actually impact this. So, for example, we have acrosome integrity, which is the little caput of that sperm. We have sperm viability. Let's talk a little bit about that in one of the studies there. We have sperm capacitation, which is the process that the sperm needs to go through before it actually can fertilize or is capable of fertilizing. We have other proteins that are coating the sperm outside or they're inside of that sperm. And we may know or may not know what the function of those proteins are. Epigenetics, so basically methylation, and there's a lot of different things. The metabolism of that sperm. MicroRNAs are very, very small molecules that are within that. They can actually regulate protein production. So, and we know that the sperm delivers some certain microRNAs within the oocyte that impacts development. Reactive oxygen species or ROS. So we know there is a certain level. It is necessary for the sperm function, and it's important that it goes up a little bit after ejaculation for the sperm to maintain function. But if it's get too elevated, it's also bad because that is one of the things that can cause the DNA fragmentation that I was talking about. So it can go there and cause DNA damage. It can also impact the cell membrane that will impact viability. So we have all these pieces of this puzzle. The issue we have, and if Lacey, if you don't mind moving to the next slide, is we don't know what is actually enough of each one of those. So we know that the ejaculate must have enough of all those desirable characteristics for that bull to be considered high fertility. The issue is we don't know what is enough for all of them. So for example, motility and morphology, we know we have a threshold, but all these other, we are still battling, trying to figure out where is that enough. So some of the techniques that I have employed and other scientists have employed, trying to understand and develop new fertility markers, will involve microscopy. And usually microscopy is one of the first steps because it's a lot cheaper to develop. And then we're going to look at sperm in a microscope. We're using fluorescent probes or using antibodies to do immunocytochemistry. The issue with this is we can only look at a couple hundred cells efficiently, right? So the way we do is we look at something in a microscope, and if we find something that it is worth exploration or continued exploration, we transition to flow cytometry, which allows us to look at 5 to 10,000 cells very quickly in one single sample, right? The more recent, we actually been employing artificial intelligence within those analyses that allows us to actually separate things that with the human eyes we are not being able to separate, but the machine can pick small nuances of changes there that allow us to separate. But all of that is still within research. We haven't been able to most of those artificial intelligence, especially some of the flow cytometry, like viability, DNA fragmentation, all that is measured through flow cytometry very efficiently. And we have some commercial use, especially for AI. Unfortunately, that is too expensive to be out there in the field for our veterinarians to be using for diagnostics during our breeding sounds exam. And then lastly, we have what we call omics, or that will involve our what we call proteomics, genomics, several of those omics that allow us to look at very, very molecular portions of the sperm with extremely high throughput data. And also we can use multiple of those together. And we're going to touch a little bit more on the omics in a few slides here. But actually, I actually have done some studies with flow cytometry that I want to share with you. And this is just a few of those that basically have the most interesting results to share. One of them is viability. The other one will be associated to the sperm capacitation that I mentioned. And actually, we saw that the intracellular zinc of the sperm is associated to the moment within the capacitation cascade that sperm is. So if we look here, that those green sperm right there on the top right, that signature 1 sperm is the sperm with the highest fertility potential that is not capacitated. As the sperm enters the capacitation cascade, it's basically a commitment. So either you finish capacitation and fertilize the oocyte, or it dies. There is only 2 ways. So as it transitions to a signature 2, which is the beginning of that capacitation process, we can see a relocation of that zinc within. So the green represents more zinc. So it relocation towards more the head and the meat piece. As the sperm is fully capacitated, we're going to see that signature 3, where we have only on that meat piece of the sperm. And then on signature 4 is basically that sperm has fully capacitated and actually died after that, and there's no zinc intracellular. Okay, so we look into these populations of sperm. And also, I focus on this CD9 protein. The main reason I want to focus on this protein on this study, so we knew that this protein is present in the oocyte. And actually, the oocyte, when don't have that protein, it actually is infertile, so you cannot fertilize. Fertilization doesn't occur when the oocyte don't have. And we had a previous study that showed that the sperm had it, and a few other reports in the literature. So I was like, okay, let's look into this protein more in details to figure out what's going on. So we actually, if you look at the next slide here, in this table, I use actually the same bulls from that previous study that I show you from the large field trial that I did in Brazil. I still had semen from that same study available. And we had bulls A and B, which are high fertility, bulls C and E that are low fertility, and that bull D that is intermediary. Interesting for us, we could not separate semen quality-wise that bull D from the A and B in any of the analysis we did. So he was always grouped the same way with A and B, so with the high fertility. So that's why it's in blue here. This is the average of several time points, including a 24-hour time point. Okay, but what we saw here was that for viability, and it's not surprising, right? Those bulls with high fertility actually have greater percentage of viable sperm average of all those time points compared to the low fertility animals. And the zinc signature too, so the 1 and 2 are the highest fertility potential sperms within that population. So we look at only the 2, and then the 1 and 2 combined, or those are the ones that were significant difference between our treatments or bulls here. And we again noticed that those high fertility bulls had an increased percentage of this high fertility population compared to the low fertility. So what we need to do, and we're working on it, is progressing here and adding more bulls, right? 5 bulls is a very limited number. So we're going to continue adding numbers and looking into this bulls or different bulls to make sure this is actually something that the industry can start applying. And lastly here, that CD9 population, we look at 4 different populations, right? If they had that protein, or if they didn't have that protein, and then if they're alive or dead. So we have live with, live without, dead with, dead without. So that what I'm showing to you here is that population positive for that protein or with that protein. And this is the funny thing, because here is the dad telling us a story. And the reason it matters for us here is because we had that such long incubation for this trial, and this is the average for all those time points. But on the contrary, for what we thought, that we were expecting to see, because in the oocyte is required, we expected high in the bull to be better, but in reality we saw that when you have more percentage of sperm, that sperm positive for CD9 in that population, we actually had lower fertility, which was very intriguing. I'm not fully sure, I'll be very honest, how to fully explain this, but I think it's very interesting, and we're still thinking about further studies to actually look what is the relevance of this. So as I said, with that omics stuff, right? So genomics, epigenomics, transcriptomics, proteomics, metabolomics, lipidomics, and other omics that we may come up with later in the future, all of those we're looking at more molecular stuff within that sperm, right? And we can actually combine several of those that will give us what we call a multi-omics approach, that give us a more holistic approach or understanding of what is going on with that sperm. So there is to date there is only 2 studies that look at actually variables that evaluate a multi-omic approach to evaluate bull fertility. And the first study there, the Telluri, they look at transcriptomic, proteomic, and metabolomic. And the Costas look at genotypes, sperm DNA methylation, and small node coding RNAs. What overall between the 2 studies, what actually they saw was that the metabolism was likely involved in regulating, it was highly associated to the regulating that bull fertility, which is very interesting for us. And looking at the microRNAs, there's some that are predictive of bull fertility. Of course, one thing is dairy bulls, and 2, they have we always fighting the limited numbers of animals, so we really need to repeat those studies to make sure that it's not something that if I want to study that is not repeatable when we start having more animals, right? But some of the microRNAs that they saw that were associated to fertility were associated to cell differentiation, embryonic development, and fetal development. So really, if we go back to some of what I was talking about early, and that really the period where we lose most of the pregnancy, right? From fertilization to about 21 days of pregnancy. Okay, what is happening there? A lot of cell differentiation, that embryonic development is happening, and beginning to start some of that fetal development. So it's really crucial, and it seems like these microRNAs are really regulating that early development of that embryo post-fertilization. So giving us more insight that the sperm is more than just delivering that DNA within the oocyte, but is also regulating more things within it. So more recent, actually, I had a student that have been working in my lab, and we started looking more in this metabolomics, which is a way to evaluate the sperm metabolism. As we look into the literature, a lot of the studies, or the large majority, if not all of the studies we have available on bull fertility, is using dairy bulls. And the reason for that is because we have so much data available because of reporting. DHI is one of them, right? So they are able to collect a lot of data. And if we think about the dairy industry, 90+% of dairy cows are artificially inseminated. So we have this AI center bulls that we have been sending semen, they're reporting. So we have tons of data. With bull fertility, we are not as fortunate to have this such of database. However, we're very fortunate to work with select sires for this study, and they actually started compiling a database for beef bulls. In part, we're very fortunate for the beef on dairy that sometimes we like to, we don't really like the idea, but it's been really good for me on the male fertility side because of that. They've been reporting the fertility of beef sires, and we're able to generate this population where we have low fertility bulls. They're really low, so the average fertility there around -4, and 0 being average fertility, and our high fertility bulls being 2+, right? 2 standard deviation of 2 deviations from what is the average fertility. So we got semen from these bulls, we process it, we separate only viable, only live sperm. Okay, the reason for that is, okay, we know that that sperm will have a bunch of junk inside, and we did not want that impacting our ability to differentiate what really is happening when they fertilize. So we got only sperm that had potential to get to the site of fertilization. We check it to make sure they pass. We are actually improving and getting viable sperm. We're above 90% of those sperms being viable, so the large majority. Then we kill that sperm because we want to freeze the metabolism of that sperm right away. And then we send it to a laboratory to do metabolomic analysis. And very, very interesting there, we start seeing some metabolites popping up. But on the contrary for what we were thinking, we were expecting to see something, okay, this is a marker of high fertility. But in reality, what we start seeing is, okay, these are markers of low fertility. So we have N-acetylglutamate and N-acetylglutamine, both of them showing a higher expression or higher concentration within our low fertility animals compared to our high fertility. And then when we put multiple of those metabolites together and start comparing them, we actually are able to separate those low fertility from the high fertility bulls. So it's very, very exciting for us in our lab to look at that. And then I'm not going to go into too much details here on all the functions. And we saw a couple other metabolites associated to fertility. But if we go to the next slide, very, very briefly here, overall, what we're seeing is some of these metabolites that are negatively associated to fertility are actually decreasing the energy production of the sperm. Some of these metabolites are actually increasing the production of reactive oxygen species, which if it's above a certain threshold, which we don't know for sure what is that threshold, but if it's too high, we know it's going to start causing damage to the DNA of that sperm. And also, so with that, we are start seeing that the low fertility bulls were having what we're calling an oxidative imbalance or a disruption on the oxidative balance. So they're basically not being able to fight that increase in reactive oxygen species production because of the high metabolism of that sperm. So this is kind of some of the stuff we're seeing here with this study. And we are planning to, with the same samples, to do other omics analysis, such as proteins and microRNAs. We just haven't been able to get to it yet. But even just the metabolites are being very, very exciting results for us. So thinking about what is our take-home message here. So basically, if we look at, and then that's not only for my portion, but Lacey's portion as well. So for a bull to be high fertility, right, or if they have high fertility potential, that bull needs to be healthy, which I don't think it's new for anybody. They need to have enough scrotal circumference to produce enough sperm to be able to be breeding all the cows that he's supposed to be breeding, and also produce an ejaculate with a proportion of sperm with high levels of desirable traits. As of right now, we're talking about high motility and high normal morphology. However, we also know that that sperm population is heterogeneous, right? So there's a lot of stuff in there, a lot of different sperm in there. So when we're trying to develop a new fertility markers here, we're trying to find that those subpopulation of sperms that would be associated to producing more viable embryos, and then those ejaculates that will have more of this ideal characteristics that we know they're good for the sperm. We just don't know what would be a good threshold for us to put in there. But also within our, so to improve our, yes, you're right, Lacey. To improve our prediction of this bull fertility, we really need to validate these biomarkers that I just showed you. We know they are important for fertility, but we really need to start validating them and finding these thresholds, but also the development of new biomarkers of fertility, like I showed you with the metabolomic analysis, and further diving into E. So these biomarkers of fertility, and another important thing for it is once we develop this, they need to be easy to be performed for both the AI industry that is producing that straw that we are buying, but also for the veterinarians that will be doing that. So hopefully we can come up with something that can be done chute side that can improve our ability to detect those high fertility bulls, and understanding what is actually enough, right, for this characteristic that we are talking about for predicting bull fertility. I think that's it for what we have for today. But one thing before we finish that I want to highlight with all of this, and I don't want anybody in this panel that is watching to get mad at me, but basically everything that I'm doing here, I'm going to be culling more bulls out of your bullpen than you having more bulls to sell if we figure out what causes more high fertility, right? But hopefully, as we keep selecting for that and improving and removing those lower fertility animals from the population, we can get back to, okay, we have this higher and higher fertility animals going out. With that, I want to thank you, everybody, Julie and everybody on the Stockmen for this invite for us to be here. Very, very, very happy to be here and sharing some of this information. I'm very, very passionate about. That's most of my work. And if you allow me, well, I'm out. So here I'm in University of Tennessee. We have a bull test that we test. Actually, the Smith paper that Lacey was showing to us, part of that data was collected here in Tennessee for our bull test, and the other part was collected at a University of Georgia bull test that we've been collaborating a lot on this. And then we have a bull sale coming up soon on December 11th. And if anybody wants more information on our bull sale, feel free to contact me. I have my cell phone in there and my email, call, text, whatever is easier for y'all. And as an extension specialist, my job is to serve our producers. So it's not a bother at all if you want to call me and have a chat about cows. I actually love it.
We all do, right?
Absolutely.
Yes, thank you guys for the invitation. And if there's any questions now or later, we're happy to answer them. But we probably also know that there's a matter of time too. So Julie, whatever you think is best.
Yeah, well, thank you for sharing your information. Very interesting research, and we'll sure be watching to see how that all develops. Are there questions from the group that you would like to pose? Again, just unmute your line, and you can ask our speakers directly. I do have a couple that came in to me, but we'll go to the folks first.
Well, I guess as we were waiting for some more questions, I would say my lab, and actually I'm working in collaboration with a couple other faculty. We continue to look into this bull fertility. That's what we're looking. So not only the overweighted bulls that we continue to investigate, and we're seeing some very interesting things that soon should be out for sharing. But also we have some studies looking at other nutrition bulls. So basically thinking about, okay, we got the bulls out there with cows. Well, they lose weight. What happened? So we're investigating to see what happens and do we need to be worried about it or not? That kind of stuff. And then bull vaccination is another thing that we're looking into. We have studies being out, proposing studies there. Hopefully we can get some funding to continue investigating some of those things.
Here's a question that was sent to me, talking about the BSEs. The question: BSEs confirm fertility but not libido. Are there reliable methods or new technologies for assessing a bull's servicing ability and desire to breed cows?
That's an awesome question.
Yeah, outside of, it's always when I get that question from people, I always say, well, watch them with your cows, right? That's one of the best measures that we have. And like, unfortunately, it does take time, and your time is worth something, right? But not catching them or not observing them not breeding is expensive too, right? So observation goes a long way on that one. Sal, do you have anything to add there?
Yeah, no, that's exactly the same thing I tell people. So after the bull has a BSC, when you turn him wild with cows, just watch for a little bit. If the first thing he does when he goes out in the pasture is go find the first tree and lay down, doesn't go check cows, or if that is repeated a couple days, like you go first day, second day, third day, he's never looking for cows. He's always laying down. I mean, that might be concerning, right? Maybe just try to move him in, put him with cows, but if that's probably the only way to evaluate libido right now is by going there and watching them to see if they do what they're supposed to.
And one more question that was sent. What, if any, advancements in semen extenders are available to improve the success rate with semen for an AI type program?
That's a good question. So there are several studies looking at different extenders. Currently, most of our industry, we either use what we call an egg yolk base or milk base. So those are the two major. To my knowledge, most of our custom collection studies use commercially available with plant-based protein. Most of them will be soy protein. Use one that I can name here that I know we use a lot is the OptCell from IMV. There are several others they're as good as. We love it for research because it's clear media and we can see a lot of things. With the milk, because of the flat globules, it's a little harder for us to actually visually see in a microscope. But yes, there are studies there. Most of those, or several of those, are done in-house by our AI industry. So really, we don't have access to those things. And we do know that some little things, and sometimes it comes into the cost effectiveness of those things. They may improve a little bit, but then it's so costly to change that usually is not viable for applying it. We've done some studies showing antioxidants may improve. But again, go back to a little bit to what I was talking about, reactive oxygen species. That's basically what the antioxidant will be battling against. If you don't have an issue, if you have a bull that don't have an issue, then you're wasting your money. So my go-to is usually finding those commercially available, even within my lab when we were going to freeze something for studies, I go for those commercially available. There's a lot of in-house studies that we are not aware, and they make sure to put those things into those media that we can purchase, or semen when those companies produce semen.
Very good. Thank you again to both of you for being with us tonight and sharing your information. For those who want to digest this again, we're recording this webinar tonight, and we'll have that available on our website, so you can go back and retrieve some more of the information or whatever to take that all in. But again, thanks to you. I'll turn it over now back to Travis to introduce our next speaker.
Thank you. I just want to say thank you again for the opportunity. It was very, very nice to be here with y'all.
Thank you.
Well, yeah, I would like to say thanks as well, you know, to Dr. Quayle and Zoka for all of that information. And I hope that everybody on the call found some pieces of that helpful in their own operation that they can, you know, move forward with. So our next speaker is likely a familiar face, or at least a familiar voice to many on this call and producers across North Dakota. Especially over the last few months, Joan Zimmerman is the North Dakota Stockmen's Association brand recorder. She's tasked with overseeing the state's once every 2-year brand renewal process, which the organization is in the throes of right now, and current deadline coming up quickly is December 31st of this year. Joan joined the North Dakota Stockmen's team in 2022. Her primary responsibilities include maintaining the state's brand records, publishing the brand book, and answering questions for producers and inspectors alike. A registered livestock brand determines legal ownership, and so those brands are much, much more than just goofy symbols on the hide of an animal. Joan has joined us tonight to offer a few reminders about the brand renewal process and answer your questions to help ensure your brand is recorded properly and can help defend against livestock loss and theft. So Joan, it's all yours.
Thank you. Thanks for having me. I am the brand recorder for the Stockmen's Association, and my office, we're charged with renewing all the brands that North Dakota has. North Dakota currently has close to 22,000 brands, and we are currently at about 11,000 renewals that have come into the office. So we have quite a ways to go before we get everybody renewed. Renewal is an important part of your operation, and we work hard to make sure that we are up to date and doing our part for you. An important part of the renewal process is making sure that all of the addresses are updated, that we have, if you want to add people, children, maybe you have a new spouse, maybe you have a spouse you no longer want on your brand, as well as maybe you've had someone in your family that has passed away that's on the brand, and so we need to get that person removed so that we keep those records accurate. As he had said, the deadline is December 31st. So renewal actually started on July 1st and it ends on December 31st. All of the renewal forms were sent out about the second week in August. And so we've been getting a lot of those back, but as I said, we still have quite a ways to go. The renewal form itself is pretty straightforward. If you're not making any changes, you just need to sign, all the current owners need to sign, and then get that back to us with the appropriate fee. And we get it renewed. You'll get your card back in probably about 3 weeks. You'll get that card and letter back that tells you that it's renewed and the next renewal date or expiration date, excuse me. If you are making changes, so like I said, your changes in ownership, you're adding, taking away, we do need a few more, not more signatures, but it can be a little bit more in depth. A lot of the owners, when they sign, they need to sign with a witness. So if if you're moving towards that where you need to make that change, sometimes that's the most difficult part of the renewal form, is getting those additional signatures. So of course, with holidays coming up, we really encourage everybody to get out your little blue renewal form and get that taken care of if you guys have it. It's pretty easy. If you do it right away now, then it'll also give you time, if you have problems, to give my office a call, and then we can help you through it. And we're very, very happy to do that, walk you through it and get it going so that there's no disruption in any marketing plans that you have, and those brands don't expire. Once the brands expire, then you would of course have some of those marketing disruptions. Yeah, that's about all I have right now. I'll take any questions.
Any questions for Joan? Joan, I'll volley you one. I know that sometimes there's been some confusion, right? You talked about the changes and the signing off, right? And so there's going to be a different ownership. Talk to us about the Section 5, right? What is the brand bill of sale or the brand transfer piece? Maybe you could explain that to the group a little bit.
Sure, thank you. If when there's a change in ownership, on the renewal form, there's a Section 5, it's called, and it does seem to be causing some problems for people. The reason for Section 5, it's called a brand bill of sale, is how it's labeled on there. It's really important if there are any changes, that everybody who's currently on the brand signs in that area with a witness. That tells us that your signature is you, as well as it tells us that everyone who's currently on the brand is aware that there's a change being made and that they're okay with it. That way we're not getting anybody added to your brand or taking off of it without all of the owner's consent, which is important. Everybody has to sign off on any change that's being done. So sometimes it might take a little bit more time to get those signatures. In the event, if you have a loved one or someone who's not in the area, well, we do have signature pages on our website that you can go to our website, ndstockmen.org, and download those signature pages. So the person who's out of state or wherever they are, they can sign it, get that page back to the person who's holding the renewal form, and then all of that can come back to us. So that will eliminate having to send the brand renewal form here, there, everywhere to collect signatures. If you guys are all if they're far apart, they can download the signature pages and then get those back to us. So just a little easier way to do things instead of having to struggle to mail the papers around. But the Section 5, it is a very important part. It can be a little confusing. So again, feel free to give my office a call with any questions.
And 11,000 left to go. So there's some blue papers out there in the countryside. So 6 weeks or give or take, right? But thank you for all the work on that. Are there any other questions for Joan? If not, thank you. And remember, that very important deadline, December 31st, 2025. Our next speaker, Elizabeth, she's going to talk about some other important deadlines. Elizabeth, I'll turn it over to you.
Thank you, Julie. I'm just going to remind you guys that the North Dakota Stockman is the official publication of the North Dakota Stockmen's Association. It is our full color monthly publication that features industry news and is a great venue for your advertising your seed stock offerings. The Stockman is published 11 times a year, monthly, except our May and June issue is combined and mails on May 11th. The deadline to advertise in our publication is the 15th of the month prior, and so we're currently working on the December magazine, or I should say Julia's, but that deadline has passed, but now we'll be accepting some ads for the January issue, and that deadline is December 15th. The holidays are coming up, as we all know, and so these are important deadlines to remember, keep in mind, and then just ask us any questions, and we'd be happy to help with your marketing campaign. As a reminder, we do also offer ring service. That is a $500 fee, and you have to place an ad in our publication. And so if you have questions on that service, just reach out to myself or the office, and we'd be happy to give you some more information on that, as well as for any of the rates, availability, becoming a member of the North Dakota Stockmen's Association and getting our magazine and seeing what it's all about. We'd be happy to send you one if you would like to become a member. Julie, like I said, is your contact for the December magazine. I'm looking forward to taking that publication back over with our big kahuna in January. But for right now, if you have any questions, just reach out to Julie or I would answer some right now if you're on this webinar. But if I'm not seeing any, I will turn it back over to Travis, and he's going to talk about more upcoming things.
Yeah, we do have a pretty big list of events coming up here in the next month or month and a half, I guess. So on December 11th is the North Dakota Scimitar Association annual meeting and dinner in Mandan. Then on December 12th is North Dakota Scimitar Classic Sale and Banquet in Mandan. Also that same day, December 12th, North Dakota Red Angus Association annual meeting and banquet in Bismarck. And then the next day, the 13th of December, North Dakota Red Select Sale in Mandan. Then into the new year, January 3rd, North Dakota Angus Association State Select Sale in Mandan. And also the 24th and 25th, the North Dakota Gelbvieh Association will have their annual meeting and the Golden Rule Sale, both to be held in Mandan. So it is going to be a busy next month and a half with lots of state association events.
Yeah, sure will. We sure appreciate our breed association partners. Of course, that's how we propagate our Seedstock Council with representatives and have a good working relationship with all of those respective breed groups. A couple of things too I wanted to add as some reminders in the Stockmen's realm. I mentioned that tonight's webinar was sponsored by the North Dakota Stockmen's Foundation. The Stockmen's Foundation here coming up in early December 6th, has its once every 2 years special fundraiser called the Stockmen's Ball. If you've never been to that before, a fun night. We say a fun night of Western elegance and support for the beef industry. So it'll be live dancing, or live, not I suppose live dancing too, I suppose, live music, dancing, prime rib supper, auctions, and a really nice time. If you're interested, tickets are available. They're $150 per ticket, and will be a fun event. A fun event, but more importantly, help support the charitable mission of the foundation. And that relates to things like disaster relief, scholarships, the research programs, leadership and professional development, and others. What I wanted just to call out and to thank many of the people on the webinar tonight helped contribute to the success of one of our projects, the Out of the Ashes Wildfire Disaster Relief Program, including many of the state's breed associations who helped supplement our fund. And with other partners, the group was able to deliver nearly $1 million in direct relief to disaster victims in the terrible October 2024 wildfires that hit much of our northwestern part of our state. And so projects like this would not be possible without the great support, again, of our breed association partners and individuals. And that's kind of what our Stockmen's Ball coming up here in early December will focus on. One final note, the Seedstock Council at its meeting earlier this year, I guess, had talked about one of the issues that plagues probably not only the seedstock industry, but probably all of us as general citizens, as challenges and limitations with our U.S. Postal Service. Those of us in the seedstock industry, of course, utilize the Postal Service in many aspects for our marketing, whether it be selling or sending out sale catalogs or other direct marketing pieces. That really is something that many of us rely upon. And I know it's been frustrating at best for many because of the extended timeframes, the unreliability, and the extensive costs associated with that. And so just wanted to bring you up to speed. I appreciate the Seedstock Council identifying this as a challenge and wanting to tackle this issue. Since the group was together, has addressed this with the congressional delegation, was a supporter for a state legislative resolution calling for improvements to the U.S. Postal Service. And the resolution on your screen is one that was passed at a recent Stockmen's Association convention in Minot that illustrates what the concerns are in identifying some of those challenges. Now, I wish I could say that would magically fix the problem, but it's a start, and it's a mission that we will continue to work toward because it's certainly important for conduct rural business at the speed of commerce, and certainly need improvements in that regard. And so thank you for identifying that. With that, I think we are done with our program. I want to say thank you for all of those who have spent part of your evening with us to learn a little bit more about Seedstock related issues and being a part of this program. As I mentioned before, the video of tonight's presentation will be available on our website if you want to go back and take another look or find the link and send it on to some friends or family for a review of that. You're certainly welcome to do so. And with that, I will bid you a good night and wish you all a very happy Thanksgiving and a great holiday season and a great bull sale prep season as well. Good night.